This is the IIT final presentation by Attiya nurse and Xiaolin.
The overall goal of this project was to analyze an unknown gene sequence using biotechnology skill to make prediction of the unknown gene function. We first determined the structure of gene by creating a working map of exons, introns, and possible mutation. To learn more about the function, we analyzed the phylogenetic relationship between our gene and similar genes. Our knowledge of our gene function was furthered when we conducted PCR to amplify a mutation of our gene. Since PCR cannot tell us where gene expression was low or high, we conduct quantitative real-time PCR, which fluoresces after each cycle. More expression equals to more fluorescence. We analyze unknown gene using AB intro and homology-based model. IIT gene transcript is represented above in tan. RNA sequence data align with our transcript shown in blue using log scale from 0 to 5. The top figure shows that 4 exons were predicted in IIT, and based on best results of identified gene, there are 4 exons based on the transcript. So predicted results and actual results showed IIT contained 4 exons. MPH3 is identified as conserved domain at IIT. MPH3 is a line response gene specific to blue light. Mutation in MPH3 will disrupt downstream MPH1 photoreceptors, transduce signal that responds for phototropism. Phylogenetic relationship was observed by comparing similar species like rice and not so similar species like tomato. Our gene is matched with the gene sequence that marked in green, which is from rice. In order to understand our gene function better, we also look at the gene expression levels from different websites. There was not enough data to make a prediction of our gene response to temperature, but from the heat maps, we found that gene express highly at young leaves and young seedling. Based on our literature research and gene expression data, our gene is very important in plant structure and plant growth.
So in this lab, we did a statistical analysis of gene expression, and the box plot range for expression for leaf, root, and stem is shown below. We got the gene expression data that we measured in the lab, and here shows the box plot by using R. Young leaf, most expression compared to the mature leaf. Overall, the stem has the most expression. We came to this conclusion as we rejected the null hypothesis because t values were less than 0.05. Therefore, this experiment will affect the gene expression. For the second task, we compared stem and leaf. We accepted the null hypothesis because the t values is more than 0.05. Therefore, the experiment will not affect gene expression. Then we did DNA extraction and PCR result. The likely nonsense mutation we identified on our working map. The tryptophan W, TTG, mutate to TGA, which is a stop codon. We designed primers based on the mutant site and ran DNA fragment with PCR. The results of the Sanger sequencing were pretty clear without a lot of noise. However, mutation was not shown on our extracted DNA fragment when we aligned the Sanger sequence with the non-mutant gene sequence. The data shown they perfectly match. So then we did a new experiment. This is our experimental design. Our original design was using blue light to treat plants for 48 hours and applying red light to the control group. We hypothesized that the treatment group will express more gene expression than the control that was treated with red light. However, our plants did not go, our plants did not go well because they didn't grow. So then we changed our experiment to hormone treatment. We applied IAA to the treatment group for 3 hours before we collected the sample and analyzed them. Then we did quantitative PCR, and these are how we did the plates. So the first 2 rows is the IIT primer, and the second C and D is the housekeeping gene primers. And this is how we ran our qPCR. Then in this lab, we analyzed the qPCR results and found that the IIT gene had no significant difference between the treatment of IAA and control. The p-values calculated were as follows. IIT control and IIT treatment p-value is 0.4523. HK control and treatment p-value was 0.7481. When analyzing the qPCR results, we found that our treatment and control for IIT showed no significant difference in gene expression because the p-value is too big. The same goes for the housekeeping gene control and treatment. In some of the reactions, no data was reported back from the qPCR. This indicates that there were errors in pipetting or the seal was not correct when the reactions were run. When comparing the CT values and the CT values for the control, the CT for the control is higher, than CT values for the IIT treatment. Therefore, IIT control had a lower expression than the IIT treatment, which is expected because the IIT treatment had growth hormone. For the housekeeping genes, we observed higher expression when treated with IIA. Thank you for a great semester. This lab was a great learning experience. We definitely felt challenged, and we are glad we had the opportunity to take this lab. Again, thank you from me and Xiaolin.